Hello everyone. I hope you can hear me loud and clear. Please get in touch in the chat if there's any issues with that. I've renamed the talk a bit since thanks to the introduction from Yuma, but I've renamed the talk to variants of inferred conserve effects because we're currently writing a paper and I'd like to show you that during this talk. What is the database? It's Variants of Inferred Conserve Effects database, or VOICE, is a graph database that maps gene regulatory features and variants with similar effects on their target genes across species, and it integrates tens of billions of relationships from experimentally derived gene regulatory data on genes transcripts and expression, regulatory or coding variants, and epigenetic loci in a cloud-based database. So that kind of clears it all up, I'm sure. It's a bit of a mouthful, but I'm going to hopefully show you this graph, which represents genetic entities and can be used for scientific research into genetic diseases. Just a quick bit about who's talking to you. I'm not really a scientist or a computational scientist. I'm kind of like more of a software engineer who works with modeling code. So I worked with computational fluid dynamics, analysis and scaling on a website startup, but that was finite element analysis, which is similar to CFD, or actually a superset of CFD. And I've worked with data acquisition analysis at a non-profit, using that was working with hardware and things, and machine learning codes, like classic machine learning, for stratigraphy predictions in a geology company. And now I'm working with this genetic analysis software. And I just want to quickly let you know who JACs are and what they do. So they're a laboratory who's a non-profit biomedical research, and they empower the global biomedical community in a shared quest to improve human health. So you can imagine with that mission what we do with software, which is when we get scientific data, and in this case, in this talk, we're going to talk about genetic data. We are interested in writing and releasing papers in order to inform the community, and software products in order to back that up. So we're a not-for-profit, so they may be services generally available for scientists. In terms of where we're based, I'm actually in Portland, which is in this list, but we've got a Portland office, and I'm based in Maine, on the top right, left-hand corner at the Bar Harbor laboratory, and there's obviously some of the other laboratories listed there. And in terms of like the diseases we look at, it'll be anything to do with human health, as the kind of elevator pitch said. But an example is that we are a cancer institute of national importance, and here are the researchers holding up a slide of tissue and of a tissue which we would analyze with our microscopy. So the point where somebody like me would see that would be with microscopy imaging. And another activity that we do a lot of is we educate biology and other students. Our computational team has an intern at the moment working in AI, and we will have another one starting next year. So that gives you kind of an idea of our mission. It's sort of not entirely we're not for profit, not entirely commercial. We kind of work with different communities. And just why do we analyze data? And I will get onto the graph stuff really quickly, but just kind of the context of what we're doing is like we'll have grant requirements, and we'll be supporting products and services which may or may not be commercial, and we'll be supporting external users. And the kind of data that we integrate will be very often tabular genomic data. In this case, which I'll show to you, is all like tabular data, so CSV, tab separated, and all these files from different sources, which we parse into a graph. And I mentioned microscopy, and there's other data sets, but in this case, we're just going to talk about tabular data. The context analysis is that there are individual laboratories within JACs with different funding, different long-term goals, different postdoc members who will actually come and go from those projects, and so they'll be contributing software analysis. And then kind of a layer, I don't want to say above, but perhaps to the side is the research grade software. So that might be where we've taken algorithms that have general usage and we've kind of brought it up to a reproducible standard. If you haven't come across the acronym FAIR, it's findable, accessible, interoperable, and reusable. So that's kind of one of the mantras around our data and our services, that you know, we can reuse it in different scientific contexts. And we do have the context of commercial grade software as well at JAX. Okay, great. So I gave a talk in 21 about this graph, and I would really like it if people could say in the chat if they came. I suspect almost nobody did, because I'm sure these things roll over quickly. But if you did come, do put a thumbs up or a +1 or something in the chat. So I did talk, and I'm not going to go over all of that stuff, but I did have a slide where I said why we thought the graph was great, because we've chosen different technologies to try and hold this genetic data, and graph just lends itself naturally to the kind of thing we're trying to model. And you know, there's a whole bunch of pluses and minuses there. I'm not seeing any +1s yet. Okay. So I'll make sure I do concentrate on explaining what it's all about. So back in 2020, we started parsing some of this data. There was actually a previous project before I joined, a previous researcher joining this project. And we were building this, we were using the bulk import feature of Neo4j and building lots of public and some of our own data sources into this genetic graph. So we have I'll show you, I'll do a quick demo of the graph in a minute, but we have different entities in there, which allows you to do further research. And in 21, I presented at Nodes and explained how we were parsing this data, which was a tough computational problem. And most of that presentation is about the computational problem of getting it into this graph. And then last year, we ran through a number of important sets of variants, which are a subset of the genetic code, and will be linked on will be associated with a chromosome and a base pair location within the genome. And we were able to look at different diseases, and we've got a paper coming out which I'll talk about. And we built another version of the graph with what's called epigenetic peak information, and that took us over the limit of Neo4j community. So we had a requirement for enterprise version of Neo4j, we became customers of Neo4j at that point. And in 24, we've been using the enterprise version, and we've been designing a new version of the database to have even more information. And we've moved to something called Nextflow. I've got some slides on that. We went to, we've got a basic user interface, which I'm going to show you, which is not released publicly, but I'll give you a quick view onto that. And visited London Neo4j office and attended the Neo4j online training for our team. And if you get a chance to do that, absolutely grab it, because it's fantastic. And I wish we'd done it back in 2020 when we started on this journey. And I do think that the people who attended that have learned something for future graphs. And it's all about the design and how to optimize your graph design. That was and a lot of the guys who attended it came back saying this is a really great introduction to why graph database is a good idea. So if you're not, probably most people here are converts to it, but if you have anyone in your company that you want to show the value of that, I'd recommend that training. Then next year we want to release a paper. We want to have graph version 3 with new genetic entities, and we want to support that with a user interface, which I'm going to show you. So that means that since the last talk, we have, you've just asked a question. I wonder what are the sources of the input of data to voice. That was in the 21 talk, so you can go back. There is a video on that. You can get that on the Neo4j website. But I'll try and. So one of them is Ensemble, if you're familiar with that. One of them is GTEx, and another key one is actually JAX's own data sets from different research organizations. So that means that since 21, we've added 29 billion peak peaks and connections between peaks and variants. I think it's more like 30. We can have a look in a minute. And liftover of genomic locations. So one of the problems is some of the data sets may not have aligned genomic coordinates. So coordinates are 2 integers and a chromosome. So like a start and stop. And they may not actually be in the same coordinate system. I don't really understand why. I'm not a biologist, but you can then have, there's a code that you can run to align them so that you can get everything nicely in the same coordinate system. We use the NetFlow pipelines, which I mentioned, and we've deployed at scale using Kubernetes. So quick demo. So what I'll do is I'll go over to the database and show you the schema. It's actually quite simple, despite the scale. That's actually a bug. There shouldn't be an EQTL node. I don't know why that shows up. And so we've got genes, and we're basically looking at gene sets between, in this case, for this particular database is human and mouse. So you have homologs and orthologs between those genes. So obviously you could go here, you have to imagine that there'll be a human gene and a mouse gene, and they'll link via these relationships, which is why it goes around the circle. And then you can go, if you want to find out if a variant is linked to a gene, you can go via an EQTL, which gives you information about the tissue and things that variant is associated with, or you can go via a transcript route, and that comes from the ensemble data. And then there's also each variant is in a genomic location which interacts with an epigenetic peak. So that's like kind of gene expression data. Again, I'm not a biologist, but if I were a biologist, I could show you some of the type of things I want to search. So what I'm going to do is paste in a query here. And what we're doing is we're going from one peak to another peak. So if I just select that Neo4j query, we go from a peak in one species, in this case it's Homo sapiens, via any connection to another gene, and then via both of them are via EQTLs rather than transcripts, and then they overlap with another peak. And what we're then doing is saying we're going to limit those variants by RSID, which is like a unique ID that variants have, and then we're going to limit it by gene. And here we've chosen FOXP2. FOXP2 is well known as a famous gene for being involved with speech in humans, and there's been identified a number of changes in that gene compared to our closest and living ancestors, relatives that we think enables some of the capabilities of speech and communication. And then we're also limiting some of the epigenetic peaks by feature type on both sides. So we're looking for the same peak in both organisms. And then we're going to return it as a table of, we're going to output some of the tissue information from those EQTL links, and we're going to output the species and the gene. The genes have unique IDs as well in the original data source. We're going to output that. We'll limit to 500 because we don't want to stay here all day. What I didn't show actually before I get into that is the scale. So there's billions of connections here. So if we look at, for instance, how many overlaps there are, we can count those and return that. And if I get the syntax right. And there's 30.39 billion of those. So that's why we need enterprise. And if we look at the number of variants, for instance, we can count in 700 million of those. Or nearly 800. So that gives kind of an idea of the scale of those genetic entities. So if I go back to that query that I was running, you can see that we've traversed across this fairly large graph, but we filtered it by the variant ID, and then we've ended up with some kind of, as a researcher, we've ended up with some kind of relationship via some epigenetic peak, which has some kind of tissue information here, and the tissue information is split over several attributes, which isn't great Neo4j design, but that's kind of a future thing to fix. because they can actually, if we go across here, you can get information encoded in the file name of the ingestion, which doesn't appear in the metadata of the file. So we'd have to parse the file names and do all sorts of things. So we keep it all, but essentially at the moment you can search on all of it. So that's why there's. So that does mean if we then wanted to say, well, FOXP2 is more active in the brain, then and we want to go and search that. But that means we have to write a more complex query looking at all these properties, unfortunately. So I have one here ready. And let's paste that in. And now we have to go and look at the brain, but the brain might have different tissue values. Might be hippocampus. It might be there's one part here called stratum. So there's obviously different parts, and we don't know if the metadata contains brain, striatum, both. So there's some kind of standardization. There's something actually called Uberon. So there's also an Uberon code which attempts to stop that happening. So let's run that, and now we find that our variant does indeed. So here we were looking on the thyroid. We actually found something on the thyroid before we ran out of we ran out of our 500 rows. But because we put the tissue in, we can then limit that down and find something within the brain. So that's obviously going to then be a different result for the researcher. So just want to change tack now, and how do we get all this data into Neo4j? So we talked about the data sources, was in the previous talk, but since then we've kind of moved over to Nextflow. And if anyone's familiar with Nextflow, put in the chat +1. But I'm going to quickly talk about it. It allows you to ingest many sources and download data from many locations in a series of commands. It's basically declarative, so you can declare your inputs and outputs. So you only declare the blue things here, and then you end up with, it figures out what to run in parallel. So for us, that's really good because we want to download many sources of files, and then we want to stream them and create the Neo4j bulk input files. And we have some added complexity, which we have to store the genomic locations in H2 databases, or any database essentially, so that we can find the overlaps without holding everything in memory. So as we stream all those files, we can go back and look up if things intersect with each other. And then we write the bulk files, and then we import those to Neo4j. So a quick look at Nextflow. What I will do is, if I go here, I can run the Nextflow command that builds the bulk input files for the Neo4j database. And I've got the -resume tag because this takes 2 days. But what I've actually done is already done it here locally. So Nextflow will actually cache those results. And you can see here we go download lots of different data from different sources. And then we create a different intermediate database for all the entities. And then we parse all those files using streaming into the files. And what that looks like is, if I go here and go -h more, you can see that I've then parsed all of those EQTL and so forth data into little CSV files, and Neo4j bulk import then has a syntax of that, which is basically all the attributes and the labels and things have to be declared. And you can see some of them actually get quite big. You've got kind of gigabyte size files. So the final Neo4j database is kind of in the terabyte range. So another thing we wanted to do is quickly talk about this as a resource for science. So when I showed those Neo4j queries, they're kind of complex, and you have to kind of know something about the database, and that's not really ideal for some of our people that might want to get value from this, and some of the scientists and biologists. So we're looking at whether we can wrap this in some kind of in some kind of front end and release that with our paper. So our paper that we've got coming out is looking at alcohol use disorder and identifies variants in genes involved with that disorder, and uses the graph to then map that between the mouse and humans. And hopefully, if I've got time, I'll do a quick demo of that. But the reason we need this is, is pretty hard to write cipher, and it gives us more security options, and it's basically going to be a more scalable and secure design as well. So we actually use Spring Boot as a web service, and Neo4j have some libraries in order to talk to the database from Spring Boot. We use reactive programming model, which I'll show you in a minute, and we have then an Angular UI which is deployed into Google Cloud, and then it's a pretty low res image of the UI here, but I'll show you the real thing in a minute. If we dive into Java code, if you're familiar with Spring Boot, there's a repository pattern, and we basically create a repository which is then consumed by the controller endpoints of the reactive web service. And in our case, we're actually using the older Neo4j pattern and then passing in a consumer, and then that consumer is implemented to pass back to something called a flux object, which is part of the reactive programming model. But there is this new, what's new to us, there's a new library which is available, so we would like to move to that, but it's still reactive in the sense we pass in the consumer to the old driver connection, and we auto wire the driver using the kind of standard Spring Boot pattern. So a quick demo of that would be that we end up generating a Swagger front end for looking at things in this graph. So to give you an example, we could search for, let's just search Sapiens, and let's just choose any RSID, and it gives us back a variant for us. So this endpoint is saying, give me back a search for a variant, give me back one that matches this species and this ID, and then it tells you where that variant starts and stops and which chromosome it appears on, and so forth. So then the GUI that sits on top of that is Angular, as I say. So it's very basic, and we haven't released this as a kind of public service as yet. This is I'm just showing you work in progress for a bit of fun. But I suspect we'll get more design around this, but this is kind of like a basic functionality. So I can search for mouse. So one of the things we mentioned in the paper is that you can look up a variant. We found this variant involved in alcohol use disorder, so you can enter that variant, and then that's actually the gene which was identified in the mouse on the mouse side. And I'm not sure why it's not coming back. Okay. Let me just start that again. And then we go AP3M2. Okay, right. And then it does like a bit of checking. It says, oh yeah, that is a gene, and it is a mouse gene, and this is a variant, and it is a mouse. If we put some rubbish here, it would say no, it's not found. And I probably could do a bit more GUI design, but that's basically what it is. And then the peak is mentioned as well in the paper, so let's put the peak in, and it thinks that that's a real peak, and I assume if we put something rubbish, it would say it's not okay. And then what we do is we press the search, and then we see some kind of result which looks similar to what we saw in Neo4j, but then gives the researcher basically a CSV file that they can use. So kind of future and forthcoming work, we would like to get that paper out. It's kind of written, but we would like to also release it with some resource that I just showed you. I think longer term next year, we'd like to move to Spring Boot Reactive in the Neo4j system for doing that, rather than the homegrown one of passing in a consumer. And there is a new disk organization for Neo4j, which we believe we'd really benefit from, because it's relatively large graph. So I think we think we could get, and we've got relatively sparse usage and so we have relatively low resources assigned. So that would then rather than keeping the whole thing in memory or anything, we would always be reading it from disk when someone does a search. I mean, as you saw, it's reasonably fast, but that's because I limited it. If it was unlimited, it would not be so fast. And we do have this Jupyter Python API for it as well, which we've coded in Python, and allows researchers to interact with the same search I've just detailed as possible. And of course, I mentioned the V3 build, which will increase the different genetic entities, and the schema will get quite a bit more complex next year. So I guess in conclusion, we really like Graph Database for this kind of scientific relationship data. It's a great way of providing resources to scientists, but it's not, they probably don't want to write Cypher, or they probably, we've got to, I mean, we may well look at Bloom as well. That's another technology that we could help with making it more kind of interoperable and easier to reuse for people. And obviously we can deal with the scale with it very well. I think we found that other database technologies were hard to deal with the scale. So it's just on to the thanks and the questions. If any of my colleagues watch this, and I haven't put your name here, I do apologize, but I had a quick brainstorm before this and put down everyone I remember working with recently. So thanks to everyone who's been helping.